So I would say, for my majority of my career, I've been trying to understand, how tissues are built and developed and maintain homeostasis, in part for tissue regeneration and engineering, but also for cancer biology. And for any of those that are listening, of course, one of the ways to do that is you can deconstruct and reconstruct the tissue. And what you hear in this session from many investigators is how people move back and forth between, in vivo models and clinical models and use our 3D organoids to actually study this. And so basically it's very, sort of dynamic and iterative. And of course, we use many of the different models that are out there, including looking at people. We use mouse models. We very much rely very heavily on organoid models, the spheroid models. And we also take it down to in silico models and what have you. Now, I think no one here disputes the sort of legacy or the kind of dominance of organoid models. I mean, the whole meeting has been highlighting organoid models, and Hans Clevers really brought this to the fore back several years ago. And you've heard about a beautiful talk from Matthias on engineering, organoid models, and you can use these for cancer. You heard a beautiful talk from Andy just recently. But basically, when I got in the business, it was a very simplified spheroid model, or called the 3D, organoid at that time. And I think it would be really instructive to understand, where this came from and my sort of journey with this whole process. We also now appreciate the idea of mechanics and mechanical force. We know that, your heart beats, blood goes through your system. As you walk, you put, mechanical force on your joints and on your bones. All this is known, and as it percolates down to the tissue, we know that the tissues are experiencing, the cells are experiencing, the changes in the tension in the tissue. They're experiencing compression. This has a lot of effects on normal homeostasis and development and cancer. And so we and others have, of course, included these concepts and used organotypic models and various different materials, and incorporated in the idea of mechanics as well as soluble factors and genetic factors when we're, for example, trying to understand tissue engineering. And we've incorporated the idea of mechanics when it comes to tumor evolution, all the way from the early, lesion that actually, eventually invades, and a beautiful talk given by Andy in terms of the whole metastatic phenotype. And all you have to do is, I looked the other day, and there are literally thousands of papers this past year on tissue mechanics and cancer, and like even more on tissue mechanics and, tissue regeneration. But I think it would be good to hear how the journey can happen and where a lot of this work started, and my own personal journey. I would say of course, I have a degree from University of Waterloo, and I'm a Canadian, degree from University of Waterloo and the University of Ottawa. Okay. But I would say that what few of you understand is, and many of you yourself have experienced, is that, you're trotting along and your life is going really well, and then something crazy happens. So when I was a grad student, I was involved in this really horrific car accident when I was actually heading on a ski vacation. And I quite literally, it looked like that guy that I'm showing you here in the bed. I broke my back, my two legs, my hands. I went from going 150 miles an hour to actually not being able to do anything, and they didn't think I could walk again. And basically, I would say that, you make lemonade out of lemons. And I would suggest that that experience really prepared me for my later experiences during basically my academic career and my science career. So I think the takeaway for any of you who are still listening is that, life is what happens when you're planning to do something else. And for all of you guys that are trainees, you could take a page out of Kafka's playbook. Sort of, it's a metamorphosis is your training, and your early experiences really prepare you for what's going to happen later on. And you have to be really resilient and willing to go with that. After I finished my PhD, I joined the Canadian National Research Council in Ottawa. It's very prominent, a very, prestigious place to go. But I ended up by actually working with somebody that was kind of unpleasant. We used to call her Godzilla behind her back. And I think that was a really good kind of description. And so from there, I would say, if you're in a bad situation, what I did was I got myself out of it. But I didn't burn bridges when I did it. So that's the other piece of information I give to anybody who cares to listen to me. What I did when I was in the process of trying to deal with the aftermath of that accident and my experience with this crazy Godzilla woman was I decided to buy a ticket to drive across Africa. And I thought I had this like wonderful vision of what it was going to be like. And I started in the west part of Africa, northwest Africa, and I drove all the way down the coast and went across to East Africa. But of course, the reality of what I went through was fundamentally different than what I thought it was. And so again, another of life's lessons. So these pictures are essentially what I actually experienced. It was quite a different experience from, the sort of languishing around and lolling around and enjoying the beautiful sunsets. It was kind of intense. Basically that was another way to prepare me for science. But I would say that, I climbed Mount Kilimanjaro. This is one thing that I thought I couldn't do and I thought I was never going to walk again. And that was a life-changing experience. And I would encourage all of you trainees to think and just embrace life and experience it for what you can. Okay, so I had a great time. It was pretty intense, but it was really good for me. I then took off and went to India. And of course, at the time that I was in India, the bubonic plague broke out. And I started getting these frantic phone calls telling me to get home. But I decided instead of going home, I decided to go hiking and trekking in Nepal away from where the bubonic plague was. And so again, resilience and kind of adaptability was a really good approach to actually face that kind of real life situation. Now, basically, I had heard before I went on this trip, I'd heard Mina Bissell talk. And, this was at a time when people never did 3D models. I mean, Hinda Kleinman started with this Matrigel stuff. And Mina Bissell actually was one of the really early, advocates and people to embrace this method. And what she showed back in 1992 with Ole Peterson was that you could put normal cells and tumor cells in Matrigel, and you could see that they look different. Whereas when you put them on tissue culture plastic, they didn't. Now, most of you look at this assay and think it's kind of goofy. It's like very simple. But at the time, it was kind of mind-boggling. And so I decided I want to go to work for her. Now, I'm a Canadian, so I called myself a piece of Canadian bacon. And so I actually had a chat with her and a phone booth in Kathmandu. Now, this was before cell phones. And, in it, she told me I have to really get myself back and move to California as soon as I could because the work was really critical. So, of course, what I did was when I came back to Canada, I packed this, of course, this is not what my car looked like. My car was actually looked like a trash heap. I decided to drive from Ottawa, you could see here in Canada, California. Now, I will say my car died as soon as I made it to California, but I did make it. And they were taking bets on whether I would. So when I got there, one of the first things I learned was I started studying 2D and 3D. Okay, so, you put a cell in tissue culture plastic, it looks fundamentally different from the cell in 3D. And, if you add lactogenic hormones, the cells that are in the spheroids actually could start to turn on milk proteins, and they could make beta casein as shown here, and they can make whey acidic protein if they make a spheroid. And so happily was trotting along. And the idea at the time was that basically for the cells to live, they needed to actually, have ECM receptors. And that time integrins were like all the hot item. And so the idea was if you block the integrin interactions, then the cells would die. Okay, so I actually worked, with colleagues from Denmark, and I looked at, as you can see here on the left, we had some normal cells that were spontaneously immortalized in culture, and their tumor correlates, right? And the idea was, well, as they progressed towards a tumor, they became less dependent on the matrix. Therefore, it should be able to demonstrate at what point, because we had a whole series of these cells, at what point do these cells no longer care? Now, I got the biggest surprise of my life when I actually took these tumor cells that you see in the middle, and I used a function blocking antibody to beta 1 integrin. And I actually, when I looked at the cultures looked like they were normal looking spheroids. And I started freaking out. I thought I got everything mixed up. So I did the assay again. I got the same results. I did it another time. I got the same results. So I clearly wasn't mixing things up. And that was my first, lessons to really understand that, science just bites you. And you have to be willing to go with what it shows you, just like life. Life actually, is when you're planning to do something else, life hits you. Now, after that, I started doing a lot of work, and I had this whole series of cells that I generated from this progression model. And as you can see here is a number of pictures, and we had a number of papers that came out that really started to try to understand what the heck was going on, right? And how do you understand why you were seeing what you were seeing? When did they actually start changing? And you have to understand the backdrop here was you don't have the molecular tools then that you have now, okay? So of course I was getting ready to go for my own lab, and I was really worried because everybody then had their own little transgenic mouse. And Mina looked at me and she said, well, now, Val, I give you the gift to 3D, and there's the pig trying to fly. That was me. And I was like really worried because I thought, well, like 3D, so what? People didn't believe in 3D then. They thought I was completely crazy. I had people like shake their head, tell me, oh, cells are not like, curved. And, there was all sorts of comments that were made. But anyways, I was lucky and I was able to get a position at UPenn. So in this case, I flew to UPenn from California and started my position in Philadelphia. And I carried on, with this 3D model. And one of the things I saw was that the cells, when they were in 3D, were fundamentally much more resistant to treatments, chemo, radiation, whatever. And it had nothing to do with the fact that the cells had differences in growth. And so I spent a lot of time chasing pathways down to try to figure out what's going on. But that still didn't explain why they were so different. I couldn't find any one thing because everything I looked at was really different. Now, I actually, when I had my position in Philadelphia, I was actually associated more with the School of Engineering. Okay, I was not near the Cancer Center or the cell biology group. And basically, this is where I was, this little circle up here, and then down here is where all the cell biology and cancer guys were. And I could tell you right up front, most cell biologists and guys that are studying cancer are terrified of engineering and anything to do with math. They're a bunch of math phobes. And I would give lectures, and oftentimes when I talked about 3D and things like that, they would be like, oh, ho hum, how is it different? All the rage then was 3T3 cells on tissue culture plastic and, signal transduction. And, they had a very different mindset. Now, what I started to really ask and was really becoming very curious about is what the devil is 3D? Why is this so different? It's driving me completely mad at the time. And so I was surrounded by engineers and physicists, and they were spending all their time talking about things like viscoelasticity and mechanical force. And of course, everybody in this group here is now at least reasonably familiar with the idea that, there is a viscoelasticity to all living tissues and cells. And this is in part because the macromolecular structure of the cytoskeleton, of the proteins, and of your extracellular matrix. But at the time, it was a pretty new concept. So basically, one of the things I started to really kind of get into at that time, and again, it was kind of heretical. People thought it was, I was a bit mad. And started realizing that, the normal tissues were actually pretty soft as compared to what you see in tissue culture plastic or glass. And that when you actually start manipulating these parameters, you get a very different phenotype. And so I think the moral here for people to take away, the takeaway is, refusing to take no for an answer is really important. So follow basically what you need to do. Now, what I wanted to understand is what was really going on here. And so I was really anxious to see, because I had this feeling that tumors were stiffer than normal tissue. And I couldn't get a penny from anybody to do the work. So what I did was I begged all the people that were big transgenic mouse guys, and I said, "Well, I'll kill your mice for you." And I wrote IACUCs to get donate, to be allowed to get discard tissue, so I could do the experiments. And what came out of this was this appreciation for the fact that the tumors were stiffer, and moreover, the mouse mammary gland was pretty soft as compared to something like tissue culture plastic or glass. So it was kind of a revelation for myself and a lot of people. And that a lot of these 3D organoid models were using materials like matchdome collagen that actually had a viscoelasticity and behavior, kind of stiffness, very similar to the normal tissue in vivo. And so I had engineering students would come work with me. The cell biologists were terrified of me. They still won't come. I have only engineering students. And a guy, Matt Pezic, who now runs his own little lab, or big lab at Cornell. He's in chemical engineering. We did some classic experiments where we took the cells and we made these little acrylamide gels, and we tuned the stiffness of the acrylamide gel. And then we asked, does it have an effect on the actual organization of that little structure? And then we could start to really understand why and what was going on. And so the gist is we had this nice paper that came out in Cancer Cell. Again, we had to fight like heck. Cell refused to publish it because they said it was too heretical at the time. And lucky for me, the editors at Cancer Cell decided to publish it. And what you can see here is that at the tissue stiffness, everything else is the same. The organoid looks pretty normal, like it makes a little spheroid, it grows the rest. And once you get to a really stiff environment, it starts to become really different, starts to invade differently, won't form nice stable junctions. But again, at the time, people were like shocked that this was happening. So, basically, of course, you could see these things, but I recognized pretty quickly that, people are going to be still very skeptical unless I can demonstrate that it actually has some physiological relevance. And I decided to take it towards cancer. We have done some development work, but I decided the cancer aspect needed to be good. And I couldn't really get any support when I was at Penn to do the work. So I decided to accept a position at UCSF where people, at least there, appreciated the tissue microenvironment. Okay. And actually, I have found more acceptance from my peers and students that are willing, and trainees that are willing to come work with me there. And since I've been there, I've really sort of explored and expanded my whole sort of horizons, and really looking at, the tumor and a host interactions so elegantly and eloquently described by my colleagues in all their talks before me. And the recognition, and we've gone into, pancreatic cancers. We've now extended into brain tumors. We started with breast. We collaborate with people on many other kinds of tumors. And they all have a change in the physical properties, not only of the X-ray matrix, but the tension of the tumor cell elicited by many of these oncogenes, okay? And so basically, a couple of little things that I could show you here. When I came, I bought an atomic force microscope. I remember my husband telling me I was crazy. It's a $500,000 machine. He said, you don't even know how to use it. Why are you using it? And my comment was, I would learn. And it's now become the mainstay of our work. And you could see here that we've used it to actually do mapping of tumors. We've done this with human, we've done this with mouse. And what we could see clearly is that as you go from a normal to a tumor, you do have a change in the matrix. It is not homogeneous, it's very heterogeneous. And I can show you here, we spent a lot of time and a lot of energy trying to figure out what's going on. And, part of it is you get more matrix that's deposited. Part of it, it's remodeled. So the MMPs actually remodel the matrix. That you get a lot of cross-linking. Now, it's not so simple as you heard from Daniela. There's a lot of other things that are going on here in a tumor. But I would say, we did show some proof of concept. For example, we showed that, in a very simple kind of transplant model, that if you put in pre-malignant and you stiffen the environment, you could make them behave more like a tumor in vivo. And we showed that if you prevent the stiffness in a classic transgenic, where there is proven oncogene activation and signaling, that you can actually prevent and delay tumor formation. So does that mean everything is one size fits all? Absolutely not. You need to question things. When we've gone on now, when we've been working with clinicians and we've done cross-linking, we've developed, 8 high mass spec methods to do cross-linking analysis. And, we've now been able to see that, breast cancers, yes, they have matrix remodeling. Yes, they have a lot more fibrosis and their tissue is stiffer and more cross-linked. And of course, we can even say, well, different flavors of tumors have different kinds of cross-linking and different kinds of stiffness. And the more aggressive types have more, cross-links and more fibrosis and more integrin signaling. And you could do these lovely little, sort of distributions of the stiffness. But again, it's not homogeneous. There's little regions where it's stiff, there's regions where it's soft. A point that was really illustrated by what Andy is saying. And that elicits very different phenotypes in the tumors. And in some cases, like Danielle was saying, you can actually restrict and compress and prevent tumor growth. We know that if you have a more aggressive tumor type, we found that they have a very more elaborate kind of cross-links, and those are elicited by different kinds of, of enzymes. And so you could then envision different kinds of therapies for those tumors. This is just a snapshot that we spent a lot of time trying to figure out what's going on. And there are dozens and dozens of labs that have looked at this. And of course, one of the ways that it's affecting cell behavior are through these integrins, because they're uniquely situated as transmembrane proteins that nucleate adhesion plaque proteins on their cytoplasmic base. They interact with the cytoskeleton. They affect signaling. But of course, piezo channels, that was a revelation about 10 years ago. That's blown up. And one of the guys just got a Nobel Prize for this. So there's so many things to discover. There's so many things to think about. But essentially, it's really arguing that there's this kind of dynamic physical context to the tumor and to tissue development that had not been appreciated a long time ago, but now has become sort of old hat. Everybody and his dog is following this. Now, we've taken this from the tumor cell, and we know that it affects, the vascularity of the tumor, and it affects, hypoxia. We have a lot of studies now where we're looking at the immune response. T cells respond to the mechanics. Myeloid cells respond to the mechanics. I have no doubt natural killer cells will. The tumors send out different cues when they're in a stiff environment that differentially regulates the tumor cell or the immune cells. The metabolism is changed as a kind of current that goes through it. So this whole field is just, exploding left, right, and center. And where we're really trying to do is look more deeply at mechanisms, understand the evolution of a tumor. It's not one size fits all. In some cases that the high tension and the scaffolding can actually restrain the tumor. In other cases, it causes its reprogramming. And so the key and the most, challenge right now is to actually think about, clinical translation. Where are we going to go with this? We're not just here to play around. We want to do something with it. I would say that I've become very much of a zealot. And my argument is, if you don't think about tissue mechanics, you're like trying to cure cancer half blind. And I think we still are using as our workhorse these organoid models. But as a kind of takeaway, what I would say is that, basically, if you believe in something, you have to stick with it. It's never going to be easy. If you see something different, think about it. And I would say persistence, hard work, creativity, but above all, curiosity and awe and a willingness to look at what the data are really telling you. One could argue that my vision of myself is like some big, huge, sort of, creature that can handle anything, when in fact, I'm probably like the Chihuahua, with a big bone, but it hasn't stopped me. I would say that leadership starts with yourself, okay? You can't force anybody to lead. You can't force people to do what you want them to do. You just do it and you lead. And believe it or not, if what you're doing makes sense, you can open up a whole new way of thinking and other people will follow. And so what I feel very exonerated, because there's a lot of people that laughed at me that are now, actually working with me. So it's actually really gratifying to have that happen. And I will leave you with one thought, basically, why do what everybody else is doing? It's more fun to do something that is slightly different. It's more fun to be that little deer over here that's actually kind of figuring things out and looking what's going. Andy had a beautiful illustration about Eket Heron and, really looking at the exceptions. Why be like the rest of the guys and you just go over the cliff? Why be, one of these many? Just be yourself. And of course, I didn't do this journey on my own. I had so many wonderful trainees and colleagues, and this is just a kind of snapshot of some of them. And I continue to have international colleagues. I work with people everywhere. I'm very agnostic. Okay, so I will leave it there.
Thank you very much, Valerie. That was very interesting. I know we called it a Meet the Expert talk. I think it could also have been a short history of 3D tissue culture. So right now the floor is open for questions. I would be very happy to read them for you if you put them in the Q&A, but it is also possible now for all attendees to raise their hand and we will unmute your microphone. And we'd be happy to have you ask.
If there's any trainees left out there, because I know many of you are probably tired. If there's any training, you can ask me anything. You can ask me about my life experiences. You can ask me, basically, how did I decide to do what I did? You can ask me science questions. I decided you've had a lot of science in this, these 2 days. So I figured it's late in the day for many of you. It's early for me. But, please go ahead and ask. There's some questions in the chat, I think. Or is there? I don't think there's anything in the Q&A. Oh,
there's one from an anonymous attendee. If you ever considered leaving academia?
When I actually had the car accident, or even before the car accident, but when I had the car accident, I kind of had this, at that time, I was so in love with science, I didn't see myself leaving. But then when I took the position at, for my postdoc, and I had already persevered through so much, and then I was with this crazy woman I call Godzilla, I considered becoming a dental hygienist. So, because I like doing art, and I thought I was going to be a dental hygienist so that I could work, 3 days a week and spend the rest doing artwork. And that's when I decided to take this, trip around the world, Africa and India and Asia. And while I was traveling, I realized that I love science and that I had something to offer. And I also appreciated that, that I was afforded the opportunity because I was born, I wasn't born rich. I was born quite modest and I had to work several jobs, put myself through college. But I realized I had the privilege to do it. And I came back very determined. So that was a change point for me. It was really difficult. I did face, a lot of sort of kind of life kind of challenges. And I've never since then ever decided that I wasn't going to do this. Because I realized no matter what you do in life, it's always going to be difficult. And even if I did some other job, I would probably be pretty contrary. You have to figure out who you are. And I know that I'm not somebody who likes to follow the norms. So basically, no matter what I would do, I would run into all sorts of issues. So I decided I was best equipped to do this in science. So the answer is, since then, no. You're muted. Henriette, you're muted.
I noticed. Thank you. I was just saying, removing media with a vacuum system always makes me feel like a dental hygienist in the tissue culture lab. There's another question from Virginia. What would you say your biggest challenge in your career as a scientist has been?
That's a really good question. I think that I've been my own worst enemy. I think that if I had to go back and talk to my younger self, I'd have sat myself down and told myself to take a Valium and calm down. I had, very little confidence in myself. it was kind of this, this weird, combination of excitement and curiosity and contrariness, and at the same time wanting to be accepted and loved and appreciated, and having, a lack of confidence. And so I was stressed more than I should have. I was worried about things more than I should have. So my biggest challenge has been to just let it go. You can't change what people think of you. And so my biggest challenge has always been that struggle, with my, wanting acceptance and wanting, but also wanting to do something that's different, and just being able to like calm down. And as I've gotten older, I don't know whether it's just because I'm tired or maybe after a while you realize this is who I am. I'm not going to change. Okay. And no matter what I do, it's going to be this way. And so I've been able to just let it go more. And it doesn't mean I don't feel, at times like I'm a fraud, that I'm fake and that I'm really not that smart and all those things. And believe me, I really do feel, I don't know whether that's a female imposter thing. I think guys feel it too. I felt like that my whole life. But after a while, I just kind of go, ugh, I'm tired of feeling like that. I'm going, yeah, you feel like that. Let it go, man. You can't change it. Just do your thing. Just do the best you can, man. Because I'm at this point, I'm north of my career. So my life's, I have less in front of me than behind to me. I'm tired of feeling like that. So just let it go and do what you can. Pay it forward and try to inspire people. And just, when I have one of those bad days, I don't, just go and, I don't know, go and have a glass of wine and go get a pedicure or something so you feel a bit better.
So I'm wondering if that would also be let it go, the answer to the question from Vaiba, who is wondering, you can always do more in science, apply for more conferences, do more experiments, write more papers. And at the end of the day, you wouldn't want to feel that your failures occurred because you didn't do enough. But how do you keep the balance?
That's a good question. I also had a slide that I put in and I took out, which was that, I like almost had this frantic feeling that I had to just run like a maniac because I needed to just do more and more and push harder and harder. And, yeah, it's a balance. I think, I've had a little bit of epiphany, and I think many of us with COVID, where, because I was like, are you kidding? I was like a mad woman. I was like, 1 minute I was in like, Singapore, and then I was in like Europe, and then I was in, the East Coast and the West Coast of the US. I mean, my poor husband, he's a sweet guy. And, after a while, he was like, we were like ships in the night. And also, I have a big lab and I'm trying to do too much. And I finally stopped and I went, wow, I actually like cooking. I get to see my husband more. It's okay. It's all right. I'm going to be dead, when you guys are still going. And as long as I try to do and stay true to my beliefs, as long as I try to keep doing my science the way I want to do it, it should be enough. It's kind of like you have to struggle with that and let it go. I think each of us has to do it our own way. Each of us has to face our own demons. All I could say is I can look in the mirror in the morning and I like pretty much, not always, but I kind of like who I am. I try to be as honest and as forthright and as I try to be as good and kind as I can. And I try to do the best science I can. And it's just going to have to be enough. Look at how many people here. I've heard talks, see the posters. You guys are so smart. It's like, wow, I'm so impressed. So I look at that and I go, wow, that's really good. And if I have, inspired one person to do better, that's good. So that's kind of where I'm at. So that's my answer.
Okay, thank you. Then another question. What characteristics do you believe are the most important for a postdoc? Both soft skills and technical skills.
I get applicants and they rattle off all their, their credentials. That's not what I look for. What I want is somebody who's genuinely motivated, genuinely curious, who's willing to work hard, Not because I don't like force people to do what they're going to do, but it's like, if you're curious and humble and want to learn, you're going to learn, you know. I mean, I think nobody gets a PhD unless they have a certain level of, neurons that are simultaneously firing. So you're all reasonably intelligent, okay? You could show up, you could do what you need to do. So the postdoc to me is then, are you curious? Are you inspired? Do you want to learn things? Are you willing to read and work with people? So you can't be excuse my language, you can't be an asshole. So if somebody, clearly comes across kind of like a jerk, I'm not going to hire them. But I have had people come in with very little skill set, but super motivated and excited. And, one of my top guys right now, he's not as smart as some of the other people I've had. I've had people that are so smart, they don't have it, you know, and the one who's motivated and curious. And, I have a guy who like sends me like, links to paper starting at 6 a.m. and I'm going, what's pathetic is I answer. Okay. So obviously, but he's like, oh, did you see this paper? Oh, this is really cool. What about this? And what about that? Anytime that to me is tells you that that's the right kind of Kool-Aid or the right juice. Because science is not about money. Science is actually not about fame. Science is about none of those things. Science has to be where you come in and you just think it's so cool. It's so interesting. Why wouldn't you do it? Wow. And if I could do something that actually might make a difference, that's just even better. Isn't that great? Isn't that wonderful? That's, I think, the key, because you're going to have so many frustrations trying to get money, trying to get your stuff published. Half my time I spend, I think I'm a therapist. You get reviews back from the journal, and you're like, okay, I gotta like pull the person, my postdoc off the floor, or my student, and sit them down and go, don't worry, we're gonna make this happen. I've been through that rodeo before, guys. I know we can make this happen. This is salvageable, okay? But if you love science and if you love what you're doing, then it's going to be fine. If you come to me and you say, but I'm not making enough money or this, I'm like, hey, there's room in science for everybody. We can help you get a job in industry. There's lots of things that you can do. You don't have to do, you don't have to do this. You could do other things. There's a lot of places for you to work in.
I think that also already covers a bit the next question from Laura, who's worried about younger people leaving science because of the demanding long hours, low salaries, etc. She's wondering how we could make it more friendly and attractive for the new generations.
I do think, there is this, and I think this is in medicine as well. I'm in the surgery department, so I'm like, surgeons are the classic AAA type personality, I did it, I like slept, I didn't sleep for 72 hours, and I'm like tough, and I could do, I mean, we have to let that go, okay? The one thing that, I think one has to, obviously, if you don't do the work, you're not going to get any stuff out of the other end. I do think that as a whole community, we, I think that the idea of the solo, researcher doing their thing, that's not going to keep going. That's not going to happen. A sense of community, a sense of, team science and interest is needed to actually help inspire our young people. Room for people to have some kind of a life, I've had some of the young women said, I don't want your life. You look too horrible. And I'm going, I had to learn how to slow down a little bit, learn how to calm down, learn how to look at things. I do think we need to actually figure out how, in some place like in the US, you have to like, hustle for all your salary. So it's really hard. So we do need to do some revamping. I do think that we need to give people better options and have ways that they can actually have quality of life and still do science. So I'm not quite sure what the answer is, but I'm willing to roll my sleeves up and figure out how to do that. I think COVID kicked a lot of people in the stomach. I think it really caused a drop in morale. It's been really tough on everybody. I believe that for many people, a sense of community And this is a virtual meeting. It's not nearly as gratifying as if you're in person. Come on, I'm a hug
No.
person. I know, although I know people are not supposed to hug, it's like not PC and all that, but you need human contact. Science is cool because, I've got to meet all sorts of people. I met Peter years ago. I've met, Manuel and Andy. Andy used to, come and hang out across the hall from me. Science is about people and, supporting people to do cool stuff and letting there be room for the person who's the maverick and the crazy guy and the one who wants to just kind of do their science, do their thing without killing themselves. So I do think we need to figure out how to modify and trim things back so that it's not one size fits all. I mean, I don't know how else to articulate that, but I think many of my colleagues would echo what I'm saying. It's not healthy. It's not healthy to say, oh, you have to work until like 2 a.m. like I did with Mina. Are you kidding? And then she'd be like on the megaphone or whatever. Where is everybody? It was like, 8 o'clock. She'd start calling me on a Sunday morning at 7 a.m.
That was Godzilla, I assume.
Oh, she was crazy, but she took care of herself. I just didn't learn how to take care of myself because, I came from a working class background. So, I just kind of always was very, yes, I'll work really hard. So we have to change.
With respect to the Mavericks, I think there was a next question. It's very inspiring that you say stick to your visions, but it's getting more and more difficult to fund your projects when they go out of the norm. So how did you find support in the science environment that is less and less interested?
I didn't get support for my crazy project. And I don't talk about, I have lots of crazy projects I'm still doing, okay?
Just don't tell your boss.
So, I've learned there's like science to get funded and science that you present. And then there's science that you do that you love to do that's kind of crazy maverick science, okay? So I have learned to be a little more pragmatic and figure out how to actually massage it and move the money around and figure out what to do. As I told you, when I first started, I wanted to do the mechanobiology stuff with tumors. I couldn't even get a tiny little seed grant from the UPenn Cancer Center or from the engineers. They thought I was complete, moron. So I had one, I told you, I finally, there was an engineer that he's now a big shot in Tel Aviv. And he was a postdoc working with my friend, Susan Margulies. And he said, oh, I'll do some measurements for you. But he said, I won't touch the mice. I won't do anything. So I wrote, these, ICAC for discard tissue and then went around literally with ice cream, those little ice cream cartons. And I got mice and I had an engineering student with me. And this guy, helped us. We sacked the mice and dissected, and he showed us how to do unconfined compression analysis. Okay. That's the kind of things that you do. No one is going to give you money for doing things that are really crazy, and they still won't. And you know what? People won't even publish your work a lot of times. But I have learned to be like, I'm kind of like a dog with that bone. I won't let it go. If I think it's really good, I'll keep pushing. Peter knows what I'm talking about. Peter does the same thing. Peter does really crazy stuff. Peter's really very creative. So if you believe in what you're doing, you just do it, man, because basically it's what you need to do. And don't let anybody tell you no, because then 10 years later, they're all copying you. And you know what? They don't even cite you. So, basically when we were doing 3D, people were like, oh, that you and your little 3D thing, and they laughed at me. I've watched people walk out of talks that Mina was giving. This is back in the 90s. Right. And then when Peter, with Hans did it, oh, he's like, the hero biscuit. And you're like, well, wait a second. Xena was doing, organoids from human and mouse mammary glands, decades before anybody did. But, you don't do it because you want someone to pat you on the back. You do it because it's the right thing to do. And I know it's really hard to take that in. Because basically it's human nature to want, and I know because I wanted people to say, oh, you're so smart, you're so great, and it's not going to happen, man. It's just never going to happen. It isn't. So you have to be willing to say, well, it's okay. I'm just going to do what I'm going to do because it's the right thing to do. And damn it, I've lived my life the way I want it.
Right now, you are the meet the expert talk.
Well,
and I'm not sure.
I made it this far. You know what? Ironically, I'm having an easier time getting money now. But I had to, I got one of these big, R35s. I had to put that in 5 times to get I got one of these DODs, and I remember it was the second time I got it, I kept putting it in, and some of the comments were so mean, man. I would be like, oh my God, I just got, punched in the gut. And they were stupid comments, really dumb comments, because people get threatened, right? And I just, kept putting it in. And then finally, I remember the last time I got invited to do it, I looked and I went, oh, there's a bunch of morons. I'm never going to get funded. And I just decided, I looked and I went, well, it's too much money to not try. And so I just decided, okay, I'm going to do it again. And I did it again. I said, well, I'm going to write it very simple. I'm going to dumb it down. And I did. And guess what? I got it. So it's kind of like, you're like, okay, dumb it down. Stop being so out there. They buy
I guess that's good advice as well for some of the grant writing. We're a little bit over time. We have 2 questions left. I think it should be fine to
answer them. It's
to answer them.
9:40 here.
You still have all day. David is mentioning, so we need all the support from others to help us on our journey. And what advice would you give to people earlier in their career that are shy or feeling intimidated or self-doubting?
Find a mentor. I didn't have a mentor. Find a mentor. I mentor a lot of people. I give them pep talks. It's easy to give pep talks, but I also give sage advice. And so find a mentor. Don't be afraid to contact somebody by email. And if they, here's the thing: is you might get rejections from 10 people, but you get acceptance from 1. So basically find a mentor that you can, talk to, find a peer group. Doesn't, now with Zoom, it can be anywhere in the world. Find somebody who thinks a bit like you. Find somebody who is also going through a bad time, but be practical. Don't use it as just, ranging on and whinging, because that's not very productive. If you get a rejection, I always tell people you have a week to cry and complain. You could put it on the ground, jump on it, throw darts at it, get angry, and then put it away. And then you go back and say, okay, get back on the horse. What do I need to do to fix it? So find a group, find a peer group, or find a mentor. And to give you that kind of, and sometimes, it's just I remember giving one person, she couldn't get her work even, reviewed. When all I did was looked at her abstract and her title of her paper, and I said, yeah, because look at your title, look at your abstract, fixed it. And I helped her to fix it. And after that, she was doing well. So sometimes it could be an easy fix. Other times, it's you have to dumb it down. Maybe your grant writing skills aren't as good as they could be. Maybe you're trying to do something and get funding for something that is, it's not good. Also pay attention to whatever is, sexy at the time, I was
put in the buzzwords.
Yeah, use the buzzwords, man. It's kind of you kind of go okay, hold my nose. I have to do that because basically that's how I get people on board. Technology always sells. It does. Okay. Now technology for technology, no, but technology sells, or what's hot right now, figure out what's hot, use the right thing, put it in the right way, collaborate with people. Science is harder. There's no doubt about it. It's always been hard for me because I was doing things a bit different, but also I kind of got into it when the pay line was, dropping like a lead balloon. So it's always been a struggle. Don't look at the other generation. They had it easier. Well, so what? Nobody owes you this as a job, right? Yeah. Nobody owes you. You do this because you want to do it. Be very clear about that. Nobody's going to bend over and give you everything. No one's going to award you with things. Now, there are people that get it. Yeah, good for them. They're in the right connection. They have the right political connection. Good for them.
Okay, we have one raised hand, so I would like to invite Anna to.
Show me your face.
Not sure if people can also.
Oh, they can't put their face out? Okay, okay, go
I'm
ahead.
not sure.
You're muted. Unmute yourself, or do they have to be unmuted by somebody?
EACR will unmute her. So Jen could switch on Anna's microphone.
And there is a couple more. Is there more?
Yeah, there's also one more question, but I figured I would. So there is one from Vipa. That there are now fellowships that allow you to open the lab straight after your PhD. Is that something you would advise, or would it be better to have a traditional postdoc first?
I mean, if you could do it, I was given advice many years ago. They said, Val, nothing will prepare you for running your own lab except running your own lab. And you're right. Because you can do all the postdocs you want, and it's not going to prepare you for running your own lab. And there are people, there are people who move right into, running a lab from their PhD. If you feel that you can do it and you have the right support, do it. I'm not going to step in anybody's way. Postdoc should be, if you have the right mentor, a chance for you to be protected and given a safe environment to explore ideas and to learn skills that will help you. Learn skills, learn approaches, learn the ways to think, or learning how to give talks, or learning how to, write grants or whatever it is. That's what it should be. Now, if you already think that you are there, why would somebody hold you back? Go for it, but don't do it unless you have a support group, okay? Because it's hard. It's just hard, and you know what they don't tell you? I would say, the hardest thing is training people. It's people. And I remember thinking, oh my God, I don't remember, was I really that screwed up when I was 20? Oh my gosh, I don't remember being that crazy. Of course, maybe I am, but I've had such a cross section of people, and there are times when I just look at it and I go, oh my goodness, I didn't realize, all their mummy issues they take out with me. And so I'm like, oh, okay. I had to really get straight in my own head about things and go like, wow, where did that come from? And nobody tells you that. Nobody prepares you for that. How do you motivate people? How do you work with them? When you have somebody who gets mad and starts stomping around and yelling at you, you're like, whoa, that's really crazy. Or when somebody, I've had guys come to my office and something's happened, and then they start crying, and I'm like, whoa. My friend told me get big man-sized Kleenexes. It's hard. It's hard. Everything gets thrown at you, but you know what? All my prior life experiences kind of prepared me for it, so it was easier, right? And now I'm like, I still get taken back. It's still hard. It's never going to be easy that way. So Anna,
Anna, you can unmute yourself.
Can you unmute yourself, Anna?
She should be able to.
Or maybe it was in it, maybe, or type your question, Anna.
Or indeed otherwise. But you should be able to unmute yourself. It has been enabled for Anna. And I'm not sure if Peter wanted to say anything. Yeah. No,
I think Peter.
He just joined. Joe, who's the one who's doing all the crazy experiments?
Nodding his head. I hope I gave a slightly inspiring speech. You guys know science. I could, people can blow people away with like data and facts and all that. But you want to know, if we all put our pants on one leg at a time. We all had to struggle to get where we are, and nobody gets where we are without having some support. I would say I've been extremely blessed in having a very supportive, husband. And whether you have a wife or a husband, a significant other, friends, family, peer group, you're not going to get where you are unless you have some kind of support group, because it can be really tough. And don't take yourself so seriously. We all at a certain point, I'm going to be pushing daisies. I'm going to be fertilizing some fields. So you just put it in perspective, right? Put it in perspective. and have fun on the journey because science is cool. And look at the tools you have. That's all I could say. I'll leave it at that. Look at the tools that are out there. Look at the
It's
stuff.
beautiful.
And don't tell me that, oh, everything's been discovered. I remember when I was in grad school, one of the students was like, oh, yeah, everything's been discovered. There's nothing more to discover. And I'm like, are you kidding? Look at where we've been. Who knows what's going to happen? They're going to be like explosions of ideas and findings and things. There's so much to learn. So just go with an open mind and an open heart and enjoy the journey. And so I'll leave it at that.